Tonney Nyirenda here from Kamuzu University of Health Sciences. Welcome to the fourth webinar, CoRE-AID webinar. So we have two presenters, and the first one is Professor Lilach Sheiner, a professor of parasitology from the University of Glasgow. And she'll be giving her first presentation. She has 20 minutes to do the presentation, and then we'll do 5 minutes questions. But also you're encouraged to put your questions on the chat so that then we can look at those questions later on. So Professor Lilach, this is your time. Thank you.
Thank you very much, Tony and Andrea. I'll do my best to be on time, and will certainly accept not having any question if I go over time. Just to say, I'm at the University of Glasgow at the Center for Parasitology here at our university, and I'm also part of the wonderful CoRE-AID that is running this series. And hopefully this should be working, and you should see all the wonderful movies. Right. So I'm going to speak today about the mitochondrial complexes in parasite, primarily in apicomplex and parasite and related organisms, so all together in Mysozoa. And so because I'm going to do that, I just want to remind everybody how mitochondria oxidative phosphorylation and the complex that mediate it work in general terms. So if you imagine a giant mitochondria, and this is the outer membrane on the top, and the inner membrane at the bottom, there is the intermembrane space here. The electron transport chain is a series of complexes of multi-proteins that make these enzymes, and those complexes harvest electrons from essential mitochondrial metabolic pathways. For example, carbon metabolism in the matrix or nucleotide biosynthesis in the intermembrane space. And so taking electrons from these pathways is necessary for their function. So this is a critical function of the chain. The electrons are moved between complexes of the chain, and then they are used to turn oxygen into water, and we call that respiration. And then the other thing that happens in some of these complexes is that the movement of electrons is coupled to the pumping of protons. And so the protons are now accumulated in the intermembrane space, and that electrochemical gradient creates the potential that fuels ATP synthase to generate ATP, and together the whole pathway is called oxidative phosphorylation. And so over the years, the field has identified critical differences in the composition and the structure of these enzymes when we compare the apicomplexan parasites to the host enzymes. And I'm showing here the data that we've generated in toxoplasma and some of the papers that describe them, data that shows these divergent features have also been produced in other parasites. One great example is the complexome data from Tako Kui's lab, who is also in our core. And altogether, we saw that there are parasite or specific or clade specific, so maybe beyond the parasite in some cases, proteins that we find incorporated in those complexes and either create new domains, like in the case of ATP synthase here, new interactions, like in the case of the interaction between complex 3 and complex 4, or recreating old domain that exists in other organisms, but where the proteins that do it in other organisms don't exist in the parasite and in related organisms. So I'm going to give a couple of examples for that from the toxoplasma. And so in ATP synthase, we showed that the clade-specific components that are found here under the membrane mediate hexamerization of ATP synthase, which in turn creates these pentagonal pyramids that are sitting at the edge of the mitochondria cristae, which you can see in the tomography here. And they are the one that define the very bulbous shape of the mitochondria cristae in toxoplasma. And this, we believe, is important for controlling the rates of respiration. And we've even shown that if we take away some of these clade-specific components, the hexamers and the pentagonal pyramids don't form. Instead, we see dimers, and you can see also that the cristae shape, which you see here in blue, is very, very different now. And that has an impact on the parasite's ability to survive. So this is an experiment we've done with Diego Hewitt, who's in UGA in the States, where we put these parasites that can't form pentagonal pyramid and have defected shape cristae, and which can grow properly in culture, and we put them in mice, their natural host in the case of toxoplasma, and you can see that while wild type typically kill mice very fast, the mutant doesn't kill the mouse anymore, even though we did have an active infection. So somewhere in the real life infection, there is a bottleneck where the right shape of the cristae that is defined by these new components of ATP synthase is important for the parasites to cause infection and maybe to survive. And now we're trying to understand where those bottlenecks are. So this is one example. Another example I mentioned in passing is that some of the mysozoan-specific components mediate the interaction between complexes of the chain to bigger complexes, which are termed in the bioenergetic field supercomplexes. And there we saw a very interesting case of convergent evolution in the mysozoan. So we found these clade-specific components that mediate the dimer of complex 3 and 1 complex 4 supercomplex together. And these supercomplex have been described before in the host, for example. So in this case, I'm showing the mouse one. And what we found is a very interesting case. So firstly, the component that mediates the interaction does not have a homologue in the mammalian. So in mammalian, another component mediates interaction between those two complexes. And not only there is a different component, but also the angle between the two complexes that this new component mediates is a different angle compared to the to what was described in mammalians, and also the directionality of where the complex sits next to one another is different in the parasite compared to the other organisms that's been studied so far. And if we model that into the unusually shaped cristae, the angle makes sense. It fits into the unusual shaped cristae. So it looks like there is a case of convergent evolution here. We get the same type of supercomplex in terms of the complexes that take part in it, but it is mediated by components that evolved specifically in the mysozoan, and potentially so that the complex could fit the biophysical features of the parasite mitochondria. Is that functional? So what's been suggested in mammalian is that this supercomplexation of 3 and 4 enhances the movement of electrons between the two complexes, which needs to happen through a mobile carrier. And it's been suggested that a charged patch of amino acid makes the sort of rolling of the mobile carrier across the two complexes more preferable by keeping it in 2 dimensions rather than the mobile carrier having to leave one complex and find the other complex. And so we tested if we see this same principle conserved in the apicomplexan, despite the fact that it's a different component, a different directionality, and a different angle. And we do find that we see the same type of patch, and we can model the roll around of the mobile carrier between the two complexes. So a real case of convergent evolution where a mysozoan-specific component is providing the same function, but in a way that fits the biophysical properties of the membrane of the parasite. And does that have any effect on growth? The answer is yes. If we mix the wild-type parasite with parasites that cannot make this supercomplex, then they don't grow as well. And of course, again, we're trying to understand now where are the bottlenecks where this very efficient exchange that is mediated by the supercomplex is required. Okay, so these are stuff that we published before for toxoplasma, and we were wondering, so we saw two cases now, new subunits that mediate new function, or new subunit that mediate the same function that's already been suggested before, but in a convergent way. And we wanted to know if this is true just for the apicomplexan, or is it potentially broader than that? And so we wanted to look at another mysozoan. So this is just a tree of the mysozoan, just so that you can have a feeling of where are your favorite organisms. So here are the apicomplexan, and the plasmodia are found here. And the coccidia, where toxoplasma is, are found here. And over here are the dinoflagellates, and here is Perkinsozoa. And all together, this is a group that we call mysozoan. And so in order to have a first stab at whether these new and clade-specific functions are more broadly conserved than toxoplasma, we went to look at Perkinsus, which is all the way here on the tree. And so it would be realistic to suggest that if those traits are conserved all the way there, then they might apply throughout the tree. And so the person who led this work is Andrew McLean. Andrew is the person who, through complexome study, reported the divergent components of these complexes in toxoplasma. And so he took a very similar approach also for Perkinsus. And what I'm showing you here is just a quality control for the prep, so that you can see that in the fraction that we ended up using, which is like an organelle enriched fraction, we can see, oh, can I move this away here? Yes. Yeah. We can see that, for example, we no longer have, or have very little contamination from, for example, from the nuclear fraction. We have no contamination from the cytosolic fraction, and we have an enrichment of mitochondrial markers. We also have an enrichment of ER markers, and that's because this is an organelle-enriched fraction rather than a pure mitochondrial fraction. And over here, you can see that we can detect the activity of the complexes. So the complexes are still intact. We can detect the formation of ATP synthase and the activity of complex 4 and the activity of complex 2. And this just to show you the complexity of the preps over here and over here in the two replicates that we've done for the Perkinsus complexome. And so now let's look at the data. And so from the names over in the bottom here, you can see that in the complexome, we recovered many of the new clade-specific subunits that we've reported for toxoplasma, and that's been reported for plasmodium as well, also in Perkinsus, in the case of complex 2, in the case of complex 3, and in the case of complex 4, suggesting that in general, there is a good conservation of these clade-specific components. This was also the case for the ATP synthase. So in ATP synthase, we found some 32 components in the apicomplexan, and of those, we can see 27 predicted in the Perkinsus genome, and of those, 26 are found in the complexome. And so one of the things we wanted to ask is, are any of those homologs of the components that mediate the hexamerization I mentioned earlier? And indeed, we do find 2 of those components also in Perkinsus. And so we went and looked into structural work that was performed by a former collaborator of ours, Alexei Amunds, and people in his group. So they have solved the structure of Perkinsus marinus supercomplex between complex 2, 3, and 4, so a different supercomplex to the one I described a minute ago. And within that prep, they also had on their grids other higher order complexes from the mitochondria, including ATP synthase, and they could find the same ATP synthase hexamers that we reported in toxoplasma. So we find some of these subunits that mediated the hexamer in toxoplasma conserved in the complexome, and also independently in a structural experiment, we can see those hexamers also in Purkinsus, suggesting that this function is potentially conserved across the mysozoan. Or perhaps even came from a common ancestor, even if it's not found in all of them anymore. The other thing I wanted to highlight from that structural work, that we could have some independent validation from the complexome, is this very, very new observation of a potential endogenous complex 3 inhibitor. So I won't take you through the whole detail of the supercomplex, because there is a lot of data there, and because the data still requires some controls and refinement. But I would like to draw your attention to one observation that was made during the analysis of the data. And that is that when we look at complex 3, Fay, who analyzed the data, could identify 3 sub-states that complex 3 is found in. And those states are defined by the distance between one of the key components of complex 2, which is called RIP1 here. The more maybe commonly used name is risky. And so the distance of risky from the active iron sulfur cluster center that is found inside complex 3. And so he found 3 positions of this risky in relation to that iron sulfur cluster center. And what he found is that in one of those 3 sub-states, in the place where the mobile carrier would normally bind, so that's where the electron exchange will happen, instead of the mobile carrier, we have a dimer of proteins that they named ISPR1 and 2, a heterodimer. And this heterodimer, if we model where the mobile carrier would sit, would potentially compete with where the mobile carrier should sit. And so they came up with a very novel and a bit hand-wavy hypothesis that this ISPR1-2 heterodimer serves as an inhibitor for complex 3. And when it's off, only then the cytochrome C, the mobile carrier, can sit in and transfer electrons. And so that is very new in the field of bioenergetics. There haven't been a proposal of an endogenous inhibitor for complex 3. So we were very excited to try to see if we can find any support that this really does exist in the apicomplexan or in mysozoan. And so the first thing is we went back to our complexome and we asked, in that cluster of complex 3 that visualized all of the homologs of the components we already knew, do we see those ISPR1 and 2 co-migrating in that cluster? And the answer was yes. We could see a small proportion of the ISPR1 and 2 co-migrating together with complex 3 in a native migration that created the complexome data, suggesting that some of the ISPR1 and 2 are indeed interacting with complex 3, at least in some cases. We wanted to provide some experimental analysis of those potential inhibitors, and because we can do that more efficiently in toxoplasma, we went back to toxoplasma now. And so what we did first is try to identify homologs of this ISPR1 or 2 in toxoplasma. In toxoplasma, we only found 1 homolog, so if there is a similar interaction, then it would be a homodimer. And we could model using AlphaFold the predicted structure of the toxoplasma one on the solved structure of the Purkinsus one, and we could see that they have the potential to form the same structure. So altogether, so far, in support of the idea that an inhibitor like that, if true, does exist also in the toxoplasma parasites. And so here Monica Singh joined the project, and she started analyzing the toxoplasma ISPR. And the first thing that was very interesting for us to see is that if we just look at the available data on the UPAT database, we could see that there is an upregulation of this protein in bradyzoid compared to tachyzoid. And this is in some ways in line with the idea of serving as an inhibitor, because in the bradyzoite stages, the parasites are much less metabolically active, so it would make sense to have an inhibitor for the activity of complex 3. Because it seemed to be a bradyzoite, potentially a bradyzoite important protein, and the CRISPR predicted value for this gene in tachyzoite seemed to be suggesting that is a non-essential protein in tachyzoite, we tried to make a direct knockout. And for some reason, we were not able to do that. This might be a technical issue. But so what Monica did meanwhile is she made a conditional mutant for this gene, and you can see that it's fully downregulated in her when she induces the knockdown. But then that doesn't have any effect on the parasite growth in culture, which validates what we've seen in the CRISPR screen, what has been seen in the CRISPR screen, and also validate the idea that it would be more important in bradyzoite. So of course, we need to repeat this experiment. We're going to try to also incubate the downregulation for a longer time to see maybe a growth defect starts emerging later on. And we're also trying to remake the mutant, probably this time in type 2 parasites, so that we can also look at the effect on bradyzoites. But altogether, again, an indirect support that perhaps this could work as a complex 3 inhibitor. And so the last piece of data I'm going to show you is the overexpression of the protein. So we wanted to overexpress the protein for 2 reasons. The first one, we wanted to look at the localization, and we were unable to tag it endogenously. And so Monica made a mini gene that is tagged separately and then expressed as an additional copy in the parasite. And you can see that the localization is in the mitochondrion, as we would expect for a complex 3 inhibitor. And then the other thing we thought, well, if it is an inhibitor, it would have an effect on the tachyzoid growth if you overexpress it rather than if you take it away. And so she's done some plaque assays, and this is all very preliminary. She needs to do repetition. This is data from a couple of weeks ago. But you can see that there is at least a trend that is suggesting that we have smaller plaques and slightly less plaques when we overexpress this protein in tachyzoid. Okay, so to summarize what I was talking about, I try to show you that we find mysozoan conserved components, and that at least by the data we have from toxoplasma, they seem to mediate high order formation, for example, of ATP synthase hexamer or of supercomplex 3 and 4. And in every case, we saw that this is relevant for competitive growth or for infection. So now we're trying to understand what are the conditions and the bottlenecks under which these formation really form catalytic advantages, so that we can understand when in the parasite lifestyle they are needed and see if we can inhibit them. And then the other exciting observation, at least for the bioenergetic field, potentially also for the apicomplexan field, is that perhaps some mysozoan may employ a putative endogenous complex 3 inhibitor that hasn't been seen before. And then the question in toxoplasma we have is whether this is important for bradyzoid development or survival or persistence, and if so, how? And those are the questions we are following up at the moment. And so with that, I hope I kept it under 20 minutes. I would like to take a moment to thank the people who are actually doing all the work that I showed you. I tried to name specifically some of them in relation to the work that they have done. Thank the alumni, both Andrew and Alex, who did the ATP synthase. I think I didn't say that specifically, have started their own lab recently. So those of you who are on the call or are thinking about their next career step, I really encourage you to go work with these great people. I'll thank the funder and my institutes and center, and again, the CoRE-AID for inviting me to talk, and thank you for your attention.
Tony, are you?
Yes, I'm here. Thanks, Prof. Lilach, for wonderful presentation. Any questions or comments from the audience? And so we just got a few minutes, for that. But if you've got questions also, we are also encouraged to, submit those through the chat.
I have one.
Yes, Prof. Steve.
Question. It's just also congratulations. Seems to be a very nice data. Fascinating. I'm still digesting it because in a shortness amount of time and me, I guess for like many of us, I'm not working with mitochondrial biology on a day-to-day basis, but really looks fascinating. Taco unfortunately would have probably many questions, but he had to run off. I see this in the chat. Also congrats from him, Lilach. Very naively, Plasmodia, anything, any suggestion or suspicions that you would have for, let's say, hypnozoids, etc.? Is there any chance that it could be also a mechanism operating in Plasmodia?
I think that's a great question, actually, Stefan, because that's the sort of things, the reason why we look across mysozoan is because we want to find things that could be important for all of these parasites. At the moment, we don't have a good evidence suggesting that the complex 3 inhibitor has a homologue in Plasmodium, which is a bummer because that would be really cool. But also, we haven't tried very hard, using more structural ways to find the homologues yet. The super complex mediator does have homologues in Plasmodium, and so do the ATP synthase mediators. And so one of the questions that Alex Mulat is asking in his lab is when in the life cycle in Plasmodium do those become important? And of course, the hypothesis based on Taco's data, for example, where you see more super complexes in the sexual stages, is that those things become more important when they are in the mosquito and they're more heavily reliant on a very efficient phosphorylation chain.
Yeah, absolutely. Or vice versa in hypnozoids, for instance, in terms of reactivations, for instance, which is still unsolved. And maybe, if you could potentially, trigger this at least mechanistically in a model system, that hypnozoids could be, whether that plays a role in maintenance of these sleeping beauties in the liver stages for buybacks. Yeah. Because at the moment we still have only one class of drug that acts against these hypnozoids. Interesting.
Okay, maybe we have a minute for one last question that is written in the chat. So my name is Dr. Bawa Boya. I hope I'm pronouncing it right. From the University of Abomey Calavi. Your work is very interesting and provides valuable insights, enabling zoologists to update zoology courses to reflect the plurality of mitochondrial mechanisms. So the question is, do you think that these differences in the composition and regulation of respiratory complexes could contribute to the development of insecticide resistance, given that bioenergetic targets are not universal? Maybe you can comment on that really quick, and then we can move to the next speaker.
I think it's a great question. I think before we answer that question, we would need to validate whether the extra components that we see that are conserved across the mysozoan are indeed contributing to bioenergetics in the mosquitoes. And if that is the case, then it would be good to start having a look, even at their divergent in different isolates and try to answer your question. But exactly that's the direction of movement, is if those are specific to mysozoan and important for survivability in mosquitoes, then they might be interesting targets to explore more. Andrea, I, for some reason, cannot see the play. Now it just came and went away. Oh, there we are, to stop sharing. I'm going to try to stop sharing.
Okay.
There's another question from Francis from Yaoundé. Shall I read?
I can read it. Do you think different differences in complexes could impact atovaquone resistance, especially in cytoplasmic heteroplasmy. So in fact, we've shown that at least for toxoplasma, we're now working on the plasmodium one as well. The difference in the sensitivity to atovaquone between human and parasite is actually embedded in differences in the conserved component cytochrome C and in how the helices in cytochrome C of the parasites are broken and therefore bringing in some amino acid to stack their side chains against the side chain of atovaquone. This is actually seen in the recent paper that we put on Nature Comms end of last year. So go have a look at that. The situation in Plasmodium is quite similar. So there is a difference, but it's not because of the new component. It's because of a difference in the conserved component. And it goes also for ELQ 300, and we have that in the paper too. Okay, I managed.
Okay, I think, yeah, perfect. Tony, is your connection working? Or
It's on and off. That's the challenge.
Okay.
But I think we can clap hands for Lyra. Lyra, thanks very much for your time and a great presentation that you have made here. Colleagues, you are encouraged that we should continue interacting on the great research that is happening on the network. Thanks so much, Prof. Now we'll move to the next presentation, and this will be done by Victor Asua. So Victor is the PhD student at the IDRC, but also registered with the University of Tübingen in Germany. And he will be presenting on evolution of Plasmodium falciparum antimalarial drug resistance in Uganda. So, Victor, now is your time. You have 12 minutes, and then we'll have questions and answers at the end. Victor, it's your time. Thanks.
Yeah, thank you, Tony. Yeah, it's my pleasure to share with the team currently online about evolution of antimalarial drug resistance in Uganda. Yeah, so before we really dive into talking about drug resistance, I just want to give the audience here some background understanding what actually it means when we talk about malaria in Africa. If we look at the most recent report that WHO usually published annually, particularly if you look at the 2025 report, it still shows that malaria is actually a very huge burden globally, but more particularly in Africa, where up to 280 million cases were reported, and then up to 610,000 deaths, over 95 of which happens to occur in sub-Saharan Africa. And when you look at this pie chart, the burden is not uniform. You realize that there's some countries that actually have lots of malaria, while others do not. But generally, it's actually a problem mostly faced in Africa. And when you look critical again, if you look at my country, Uganda, we happen to be one of the 5 countries really facing the scourge of malaria. And therefore, for us, it's a very big concern, and as it is the case for many other countries in sub-Saharan Africa. So, but then, despite all this huge scourge of malaria in sub-Saharan Africa, there are really important tools that helps us to cope up with this challenge. Very important among them is chemotherapy, which is where we use medicines to treat people who are already having malaria infection. And also chemoprevention, where we try to protect vulnerable populations from getting infected by malaria by giving them medicines before actually they get the disease. So that way you can keep them safe for a certain period of time from being infected by malaria. And going a little bit more into detail, for malaria chemotherapy, particularly for malaria case management, we do not have many options. There's only currently artemisinin-based combination therapies, which is used for treatment of uncomplicated malaria. When I say uncomplicated, I mean malaria which is not that very severe. And ACTs are actually a combination of two drugs. It's an artemisinin component combined with a long-acting partner drug. And then for people who already have severe disease or severe malaria, the treatment options are a bit different. Where currently we use artesunate as an IV, like intravesiculum. And this is often followed by another full dose treatment of an artemisinin combination therapy. But the bottom line here is that artemisinin play a very critical role because both in the treatment of uncomplicated malaria in the context of combination therapy and also the use of artesunate for the management of severe malaria as the initial treatment also being followed by ACTs. So therefore, artemisinin are critical here in malaria case management. Then when we talk about malaria prevention, there are also not many options. At the moment, all the malaria prevention strategies rely on sulfadoxine, pyrimethamine, and therefore it's also one of the drug combinations, drugs that I would say plays very critical role when it comes to dealing with the problem of malaria. Here I just saw the drugs that are used for malaria case management and chemoprevention. Like I said earlier, for case management, we have the artemisinin combination therapies that we refer to as ACTs. And in this group, we have around, I think, around 5 combinations. But the key here is we have the first one, let's say artemether-lumefantrine, a combination of artemether and lumefantrine, and then dihydroartemisinin piperacin, artesunate amodiaquine, artesunate pyronardine, and then we have artesunate morphloquine. And then, of course, I just talked about the IV artesunate for severe malaria. And then for chemoprevention, you can see here the different strategies that are used. The IPTP, which is intermittent preventive therapy in pregnancy. This entirely uses sulfadoxine pyrimethamine. The perennial malaria chemoprevention, which is given to young children, also uses sulfadoxine pyrimethamine. And the intermittent preventive therapy in school children relies on SP. The only bit difference is in seasonal malaria chemoprevention, where still SP is combined with amodiaquine. And then for post-discharge malaria chemoprevention prophylaxis, which is not, malaria chemotherapy, which is not very common, but for this, the current WHO recommendation is to use DP. And therefore, in this slide, you realize that artemisinins and SP actually play a very critical role when it comes to malaria case management and chemoprevention. But the challenge with these strategies, both chemoprevention and treatment, is actually the parasite's ability to develop resistance to these drugs. And if you look at this schematic, if we start, let me just find my pointer. If we start down here, if you go into the overall parasite population, you'll find parasite population that is very diverse. If you look at a genomic level, and in the whole population, there may be sensitive and tolerant parasite strains. And if an infectious bite occurs in an individual, so it's often that you may develop symptoms, and if the individual develops symptoms, and then you have to receive treatment. And after treatment, if there are some tolerant strains, they may persist after, they may not be cleared. And if they fail to be cleared, this may continue to be transmitted to the next person again. And this cycle continues. And as this cycle continues, then there is selection pressure. And that means ultimately the parasites will become resistant to the drug in question. And therefore, this makes drug resistance kind of vicious problem that will always be there because by treatment, we are kind of applying this selection pressure to the parasite population, and we have to be vigilant trying to monitor for whether there are any parasite strains that will develop resistance. And currently, for the drugs in use, we already have some key targets. For example, the ones I've listed here, that I'll be talking more about. So just to maybe bring you up to speed, when we talk about antimalarial drug resistance, there are approaches that are commonly used to try to look for evidence or presence of resistant parasite population. A key among them is to conduct therapeutic efficacy studies where you have patients who are infected and you give them medicines and follow them out to see whether they, first of all, clear the parasites and whether they remain parasite-free for a certain duration of time. And this is what WHO recommends to be conducted every 2 years, and it is required for policy decisions in terms of whether drugs should continue to be used for treatment or whether they should shift a new drug for malaria treatment. And then also, a second approach that is used is also ex vivo or in vitro sensitivity tests, where you obtain malaria parasites and then expose them drug in a laboratory setting and see whether it can kill the malaria parasites and how much drug concentration you require to kill the malaria parasites. So it gives an insight into whether there is tolerance in the absence of other host factors, including immunity, that will actually mask some of the early stages of tolerance and that may be emerging. Then third, is looking for genetic polymorphisms in the parasite gene that are associated with antimalarial drug resistance. This can only be applied if you know which genes kind of mediate resistance. And the advantage here is you can do this on large scale. And like I said earlier, we already know some of the targets for the drugs, and then actually we have been applying these to try to look at antimalarial drug resistant evolution in the parasite population, and this would be the focus of my talk. I think I'll focus more also on artemisinin in the interest of time. So, as you may remember in the title, I said we'll be looking at evolution of antimalarial drug resistance in Uganda, and particularly we'll focusing on spatial and temporal patterns of artemisinin-resistant Plasmodium falciparum. And just to give you a context, this will focus on the surveillance that we have been doing in Uganda for over a period of time from 2016 at different sites located across the country. And at each of these sites, we obtained samples from patients who are infected with malaria and genotyped them for different polymorphisms that we know are associated with antimalarial drug resistance. And one of those areas of our interest is artemisinin partial resistance, which is actually a critical problem because it impacts on how the ACTs work. And just to give us a bit of background, in 2006, ACTs were actually introduced, artemisinin combination therapies were introduced. That was when actually there was high-level crop resistance and also widespread across malaria endemic countries. And in 2008, is a few years after replacement of chloroquine by ACTs, artemisinin partial resistance was actually detected in Southeast Asia, particularly in Cambodia. But luckily now, for over close to a decade, Africa was spared. There was really no clear evidence that this was a problem in Africa until 2018, when an evidence emerged in East Africa, particularly in Uganda, that the parasite populations had actually acquired genetic mutations that might impact how they respond to artemisinins. And this also was later, like 2 years later, reported in Rwanda and now also in Horn Africa. And as I speak now, it's actually these mutations, a number of them are seen almost in the whole country, also down here in Rwanda, and also an independent emergence of these mutations was also observed in the Horn of Africa, particularly in Eritrea and Ethiopia. And so therefore, artemisinin password resistance is now a concern, and not only in Uganda alone, but now a number of countries in Africa. And if you look deeper, they actually now spread also into other areas. I think in Tanzania, they've also now published this petition, Democratic Republic of Congo. So therefore, it's a problem that is actually growing in the sub-Saharan Africa, particularly, I think, the regions in what I would say, the Great Lakes region and the Horn of Africa. So a bit of detail on artemisinin password resistance. This is actually not the classical antimalarial drug resistance where the parasite is completely failed to clear, but it is a situation where there's delayed clearance. If you look at this figure here, I picked this from a paper published in 2008. I think this was the first report from Southeast Asia. What you see here is parasite densities on the y-axis, and then time after treatment on the x-axis. And then we have here 3 curves. The first one is the curve for the treatment failure in blue, and then the one for treatment failure due to artemisinin resistance, which now we call password resistance, in yellow or orange. And then the green is the one for the person who cured. So clearly what you see here is for the person who cured, if you look at the parasite density, by 24 hours, the parasite density has dropped from 100% to 10%. But for the one who failed, by close to 40 hours is when you achieve a drop from 100% to 10%. So there's kind of like a delay in parasite elimination rate. And this has been in the deep analysis, it was actually shown that for the artemisinin password resistance, particularly in Southeast Asia, was associated with parasite clearance of around 5.5 hours, like clearance half-life of 5.5 hours when patients are treated with ACTs. And then when there are also lab-based experiments, which we have developed to try to understand this phenotype, which is where the parasites are exposed to a high dose of DHA, that's dihydratomycin, which is actually a dose of around 700 nanomoles, I think. And for those parasites with delayed clearance, like around 1% of the parasites, or more than 1%, actually survive exposure to 6 hours exposure to very high dose of DHA. And therefore, these 2 phenotypes were used to define artemisinin password resistance. And like I said earlier, these phenotypes are associated with mutations in a gene called K13, particularly in a specific region called the propeller domain. And field studies have actually validated approximately 20 or even more of these mutations in different malaria endemic countries. So in Uganda, we started the surveillance in 2016. What you see in this map here is actually spatial and temporal prevalence of the different K13 mutations. The ones I've listed here, at least, are the ones we know are associated with delayed clearance. And all these were actually we detected in our parasite population. And what we clearly observe here is that from 2016, the initial mutations that were present in the local parasite population were the first 2 here, and this was mostly in northern Uganda. And as time goes, their prevalences continued to increase, and then they also continued to spread to different parts of the country, to the extent that by 2022, you see this in almost all parts of the country. And then also another independent mutation, also like the third one here, also we observed it mostly initially at one site in southwestern Uganda, but also for it has remained kind of localized in southwestern Uganda with some kind of random observation, detection in 2021 here, but it mainly remained localized in southwestern Uganda. But however, we also see that the prevalence also in southwestern Uganda continued to increase from 2016 to 2022. Another one, 561, this was originally reported by a study conducted in Rwanda. And from 2021 and 2022, we also see it down here in the southwestern part of the country, which is actually right border in Rwanda, which potentially shows that this was a spread from Rwanda maybe to the southwestern regions of Uganda. The last one I saw here is 441L, not yet fully studied, but from our molecular epidemiology study, it kind of appears to be polymorphism that is also under some positive selection, because we see that initially it was seen in southwestern Uganda, but over time its prevalence also appears to be increasing, and we appear to see it at more sites by 2022. So in summary from this slide, we see that there are multiple emergencies of artemisinin, parasite-associated K13 mutations, at least 5 of them we can say in Uganda, and almost seen in the whole part of the country. Earlier on, we actually did a deeper study to try to understand the first 2 mutations that I talked about, the ones in position 469 and 6756. This was trying to understand their relatedness. And for this, we did microcellular genotyping for those which are flanking the K13 gene, and then analyzed parasite relatedness and try to see whether these parasites are all from the same genetic background or whether they're independent emergencies. So here we have 3 mutations of interest we are looking at. The 469F, which is I don't know whether this is red, but at least here it's the one. And then the green 469F, and then the blue 675V. So what we see here is that if we start with the 469F, it appears to be one population of parasites, or like I said, clustered in the southwestern part of the country. But when we look at the 469Y also is also potential like a single emergency. And this also, if you look at the names of sites here, are also all sites in northern Uganda. But what's a bit different is for 675V, there appears to be, first of all, one population here, which is in sorry, I think I got disrupted. Yeah, so I got disrupted by a phone call, sorry. So if you look at the 675V, we have this population of northern Uganda here. But again, there's also this other one here. When you look at these names, these are names which are far away from northern Uganda. So it is possible that for 675V, there are like more than one emergencies. And here we look at whether any of these parasites may have been imported by comparing them to parasites from Southeast Asia, the ones which are carrying the different K13 mutations. So they all group far independent from Southeast Asia parasite populations. Clearly, these are local emergencies in Uganda rather than any sort of importation from where they were originally detected in Southeast Asia. So in summary, we can say that there are multiple K13 emergencies that do show temporal increase with dispersal spread. And of course, for the other 2, they are likely to be independent single emergencies, but for 675V, it's possible that there are like more than one independent emergencies happening in Uganda. Also, I just want to show also something briefly on aminoquinolines, as we know that they are also, yes.
Victor, sorry to interrupt you. Could you please wrap up?
Yeah, if there's
have time for questions. Thank you.
Yes, if we are out of time, I think I intended to most talk about K13. I can skip these 2 mutations. And yeah, we can go into questions because I anticipated that by 10 minutes, the K13 latest story will have taken all the time. Yeah, thank you. I think we can close here.
You can
So
finish
back
your
to you.
acknowledgement, but yeah.
Yeah. Thanks. So yeah, many thanks to the support I've received from the different grants and also my supervisors. Yeah. So yeah, thanks. Back to you, the chair of the presentations. Yeah.
Hi, Victor. Well done. I think a great presentation, very important, particularly in the regions that you have actually done this. And certainly, I think this should also be applying to even other regions like Central Africa and the like. Yeah, there is a question on the chat. I think we could actually start with that. The question is from Francis Nkengo, and are there other enhancers of
Hello. I can't hear the chair. I
think
hope
you
I'm
can
still
read
connected.
it because Tony's connection is a bit unstable.
18 mutation?
Can
Maybe Victor, you
everyone?
can also read the question.
Mhm.
So there are 2 questions from Francis. Do you see it? I can also read this otherwise.
Yes, I trying to find how to. Can everyone hear me? If you could just read
Yeah.
maybe for me.
Yeah, we can hear you.
Yes. So yeah, interesting from Francis. The first is, are there other enhancer mutations co-driving the spread of these multiple administration-resistant mutations in Uganda? I don't know if I do understand you well when you talk about enhancers. Do you mean I'm not sure if I understand your question very well when you say enhancer mutations. But otherwise, we do think that partial resistance is driven by K13 mutations. There are also there may be potential other mutations also that may mediate delayed parasite clearance. That I'm not sure if I understand your question when we talk about enhancers. Maybe you may want to add one or words to clarify on that.
Maybe Victor
Yes.
Francis can also explain. He's there. Francis, maybe, but I guess he meant compensatory mutations by enhancer. Enhancer is also difficult, but maybe compensatory mutations, for if there were any fitness costs associated with the K13 mutations, whether there are compensatory mutations, whether you had identified any that would compensate the potential fitness cost carried by those K13 mutants.
I'm not sure if I would say they are not there or they're there because we haven't investigated in detail whether there are specific mutations that do occur with the K13 that would enhance or give it some fitness advantage. What we know is in the Southeast Asia, what they did is what they found is there are some mutations that they are likely to be like background mutations, like founder population-related mutations, but there is no clear evidence that they actually provide some compensatory advantage. Also, we think that yes, it's possible that genetic background may be important because also not all parasites that carry K13 mutations actually experience delayed clearance. But it's also the caveat here is the background genetics is so complex. Therefore, they are not clearly defined compensatory mutations that may play a role in enhancing fitness for K13 mutations. So it's a wide field yet to be studied. There may be multiple of them that may actually play a role, but yeah, we're not sure which ones do. Then if I haven't answered you, you may still go ahead and answer if you have more questions, but also somebody has a question here, whether these lead to treatment failure. K13 mutations alone actually do not lead to treatment failure, but what they do is, what happens is that if there is also resistant to partner drug, combination of the two actually lead to treatment failure. Delayed clearance in combination with the also loss of efficacy to the partner drug leads to treatment failure because, as you know, ACTs are a combination of drugs. Then from Francis, again, interesting to see disparate geolocation of K13 mutation in different regions in the same country. Are the drug combinations different? No, the drug combinations are the same. We use AL at the metal fountain throughout the country. Then from Akombe Mote from Cameroon, working on HRP2/3 deletions. Could you explain the method used to detect spatial and temporal patterns at missing partial resistance, particularly how clinical parasite clearance data are integrated with molecular markers like K13? The clinical data is obtained from therapeutic efficacy studies where we do measure parasite clearance rate by obtaining dry blood spots after every 6 hours to estimate parasite densities, and that data can be used to estimate parasite clearance rate. And then the K13 information or the K13 mutations are usually obtained from genotyping. You can use Sanger sequencing. If you have amplicon sequencing, you can also use amplicon sequencing. And then you can integrate the two data sets and then try to understand how the two correlate. Then any evidence on non-K13 independent mutations in your parasite genomic data. This is what we are yet to investigate with the data set we have. Yeah, thanks. Very nice question.
Okay.
Yeah.
I think there are no more questions in the chat. There's one question from Stefan. Okay, Stefan, go ahead.
Yeah, thanks. Thanks also, Francis, for the many good questions. Good to be in touch again. It looks like you were hijacking our discussions here. But it's good to have many questions, as
Yes.
always. My question is a more general one, and probably, Victor, you know what's coming now. It's like for public health, and we are discussing this, but maybe you have since then had new ideas. What can we do, depending on whether this is independent emergencies or whether it's a single emergencies, and whether there would be any control strategies that would be appropriate to, let's say, more, would be better to suppress independent emergencies. But altogether, what do you think is the public health importance of, of how, in other words, the knowledge of whether this is independent or a single emergence? How does it help us to combat drug resistance or here partial artemisinin resistance?
Yeah, I think these are 2 points, considering independent emergencies versus multiple emergencies. At the moment, I think the question of single emergency is no longer, I think, a problem, because even if the single emergency, we now see it everywhere, because we have K13 mutations. Although, for example, one of the markers, it was in northern Uganda, but we see them almost throughout the country. But multiple independent emergencies is actually a lot more concerned, because it means that the parasite populations, I would say, they have predilections to, it will mean that this emergence may happen in other places quite easily, because if we can have more than one emergency in Uganda, and then the possibility of this happening in other countries is very high. But for Uganda's context, in relation to how to manage this, I think there are two aspects to consider here. The first is to protect the partner drug right now, which is lumefantrine, because if it fails, then we are headed towards a number of clinical failures happening down the road. And therefore, we need a scenario where the pressure on the lumefantrine is lessened, because right now it's almost exclusively the drug used for treatment. And if its efficacy or its potency drops, which is actually a risk, because right now what we see is it's being exposed more, because the artemisinin, which is protecting it by rapidly clearing parasites, is no longer doing that, and it's exposing the lumefantrine even more. And I think at the moment the major concerns would be to lessen the pressure on lumefantrine. And then down the road, where the options that are available is to bring in maybe non-artemisinin-based drugs, so that also we can maybe have this in the context of multiple first lines, so that both the artemisinins and then the partner drugs are protected.
Thanks.
Okay, thank you very much. I think we have to close now. There's one last question from Francis. Is there evidence of lumefantrine resistance in Uganda? If you need to go, also feel free to go, but also if you want to keep listening to the answers, feel free to stay.
I don't know whether I would say lumefantrine resistance, but we have clearly seen that over time, the IC50s of parasites in Uganda to lumefantrine has doubled. I'm not sure if whether to call that resistance, but there's evidence that if you look at 10 years back, the geometric mean of IC50 to lumefantrine, it is coming to double to what we see now. So clearly there is developing tolerance to lumefantrine. For sure, I don't know where the cutoff for resistance would be. That's best I can say, Francis.
Okay. Yeah. So thank you again, Victor, for presenting today. This was a very fascinating talk. Also, Lilach, thank you very much. She had to leave a bit earlier, but she also sent congratulations to you for this very interesting results. And thank you to everyone for joining today. It was very nice to have enough time for questions and interactions as well. It was a very nice discussion. So I invite you to stay tuned to the updates. I will update the calendar for the webinar for the year. And also I will see you again in a month on the 3rd of March. Remember that the webinar is on the first Tuesday of the month. And I will notify you when the recording is ready. And have a nice rest of the afternoon and have a nice week. And thank you very much. Bye.